Hello and welcome to this webinar to introduce the ABX Guide, an open access antibiotic treatment guide focused on pathogen-directed treatment for resource-constrained settings.
This is the first of several webinars, and this is the global launch. Let me start by introducing myself. My name is Souha Kanj. I'm a professor of internal medicine and infectious diseases and chair of the infection control program at the American University of Beirut Medical Center. I am president-elect of the International Society of Antimicrobial Chemotherapy. This is the program of today's webinar, and we have 4 speakers who will cover various parts of this webinar. And please, throughout the webinar, if you like to ask any questions, just type them in the chat area, and we will address them during the Q&A sessions. As president-elect of ISAC, I would like to share with you a few introductory slides on what is ISAC. So ISAC is the International Society of Antimicrobial Chemotherapy. And this is a society that was founded and is in 1961 and is a federation of over 100 affiliated member societies. The aim of this society is to increase the knowledge of antimicrobial chemotherapy and combat antibiotic resistance around the world. We have a board of directors and we have an executive committee. And as you see from the names and the pictures, we have international representation in the board of directors and in the executive committee. We also have ex officio members, and we have executive office with Fiona, the CEO, and Fee Johnston, the executive officer. And here I'd like to thank Fee for all her efforts in really planning the series of these webinars. The International Congress of Antimicrobial Chemotherapy, or ICC, is the conference that the ISAC plans every second year. And as you see over the years, since we started in 1959, every other year, the Congress has happened in various parts of the world, including many countries in Europe, in North America, including the US, Canada, in Asia, and more recently, we've had the ICC Congress in 2019 in Dubai, and the last prior one was in Perth, Australia in 2022. The next webinar, the next ICC will happen this year in November 2024 from 3 to 6, and it will happen in the beautiful city of Istanbul, and this is in collaboration with the Turkish Society of Hospital Infection and Control. ISAC has 2 peer-reviewed scholarly journals, the International Journal of Antimicrobial Agents and the Journal of Global Antimicrobial Resistance, with the editor-in-chiefs being Jan Lee and Stefania Stefani, respectively. ISAC also runs an academy. The academy website was launched at the start of 2022. It's an online educational resource which hosts both ISAC have e-learning modules on antibiograms, respiratory infections, and fever in returning travelers, and others that are being developed. And also, ISAC Academy plans various webinars with global expert speakers and global audience covering up-to-date issues in infectious diseases. Now, the Alliance for Prudent Use of Antibiotics was acquired by ISAC in 2019 after the retirement of its founder, the late Professor Stuart Levy. And as you see, APOA has been really very active at many fronts. We have developed at APOA several webinars. We have periodic newsletters on very important topics of infectious diseases. Other examples of APOA activities include the antibiotic cards that have been developed. and position papers by APOA board members on important topics in antimicrobial use and antimicrobial stewardship. Now, for all people who are joining this webinar, if you have any comments or feedback, please feel free to share them on our social media. We have LinkedIn, X, Facebook, YouTube. All feel free to really, if you have any suggestions, to email Fee Johnston at this email, secretariat@isac.org, for any ideas for future collaboration or feedback. Now, I stop sharing because I'd like to really introduce the man behind the ABX guide, and that is Professor Heiman Wertheim. He really came up with the idea, and he's the engine behind developing these guidelines. Heiman is a professor of clinical microbiology. He's the head of the clinical microbiology department at Radboud University in Nijmegen in the Netherlands. He's also the president of the Dutch Society of Clinical Microbiology. And as of November 6, 2024, he will assume the role of president-elect of ISAC. Heiman will be talking to us about the why of the antibiotic guide. Heiman, the microphone is yours.
Thank you, Souha, for your kind introduction. I hope you all hear me fine and can see my screen. So I was to briefly sort of introduce you why we started with the antibiotics guide and why ISAC thought it was a good idea to start to fund this endeavor. So it's really thinking about, it's really everybody agrees it's important to have up-to-date guidelines. If you see many recommendations in the literature, that often also what you see in low resource settings, actually there are no up-to-date guidelines, and particularly not in a directed therapy. If there are directed therapy guidelines available, they're often behind a paywall, or they're really sort of more tailored towards high income settings, for instance, the American setting, which is not always the right sort of context or the right approach for people in low resource settings. And of course, low resource settings have also various needs. So an African setting will be very different from an Asian setting, but also within Africa, of course, there's also a lot of differences. So if you think really about it, you have now the AWARE Handbook, which is a great initiative, and Mike will talk about that later, is really an empiric treatment guideline with a lot of details and also looking at what the potential etiology is and why a certain empiric treatment is chosen. But really, sometimes you really need to have diagnostics and really to know the best treatment for this patient. And also what we see, there's a huge dynamic going on with carbapenem resistance. You have many different kinds of mechanisms around resistance, particularly for carbapenems, for instance. And it really matters to know what is the mechanism, what is really causing the disease. And it's also changed so rapidly. How can we help people if you know what is causing the disease? How do you best treat it, for how long, and what dose? So therefore, we thought it was really quite crucial to develop this antibiotics guide to make sure it's also freely available and up to date, but really as a directed therapy guide. So it's not, so it's really complementary to the AWARE Handbook, which has been developed by WHO, which is really empirically focused. So in that sense, we think it's really a nice addition to the AWARE Handbook. Of course, a treatment guideline is never a thing on its own. And if you have the guideline, you're ready to go and everything will be, the great care. But it's really a crucial component of standard of care. But it was a really nice paper in the PMJ Global Health for a couple of years ago. That is really that the clinical practice guideline provides a standardized and systematic approach to responding to disease, including the treatment. But you have to, it's really about the context. So you should have a functioning health system with trained, well-trained, adequate clinicians, nurses. You need to have availability to the drugs, but also to the diagnostics or your microbiology, and to have a supportive environment. And the paper comes with a really nice sort of figure with the whole context that you need to have good resources of human resources, knowledgeable people. So not just having available to tests and drugs, but really have people around who can think about it. besides just having the guideline, then your lab support, for instance, but also imaging, but also a good pharmacy. And then you come to the next point of having the guidelines, but also having a whole feedback loop around it to make sure it can change. And the idea is that the antibiotics guide is really something like a starting point that people don't have to reinvent the wheel. So it's really accessible, but we also want that people can also tweak for their own setting if, for instance, a different kind of drug is available in their setting. An important point is what people always tell us when we started with this project is that, yes, but if you have direct therapy, you need diagnostics. So how can we use it? And this is exactly what we mean and why we think this is important, because a directed therapy guideline shows you that diagnostics are crucial and allows you to go to your leaders in your hospital or in your country, to your government, like, look, knowing what is causing disease really matters, and it really matters what we give the patient. I mean, this way we can actually improve patient outcomes. So in that sense, we want to also use the antibiotic guide as a way or as a means to show the importance of diagnostics, because only if you know what you're treating, you can be directed. Otherwise, it doesn't make any sense, and the AWARE handbook is in that sense sufficient. So in the end, we have to think about what kind of drugs are we choosing. So it's quite important to think about, okay, for low resource settings, not to think only about the most novel and costly drugs, but the concept is to be as narrow as possible and at acceptable cost, but in a way that's also acceptable for a low resource setting. Hind will talk about it more later on in this session. So in the end, we want to know, okay, what is causing the disease? Where is it? Does the antibiotic reach it? What are the resistance mechanisms behind it? Because that can limit your choice. What is the location of the infection? Can this antibiotic actually reach that point? Thinking about meningitis, for instance. But indeed, you have to think about the access of antibiotics. Do we have them available? And if not, can we talk to the leaders, to the pharmacist? Look, we really have a problem with this infection. We need these newer drugs, or even sometimes the old forgotten drugs, to be able to treat these infections. And allergies is, of course, another thing that limits your options, and you need alternative strategies, which we also provide in the antibiotics guide. So here's just an example of carbapenem resistance, so CPE, so carbapenemase producing Enterobacterales. Just an example of a paper which shows you that, for instance, in the boxes below, so you have a suspected carbapenem resistant infection, but you see that, for instance, for ceftazine avibactam, it doesn't have any activity against metallo-beta-lactamase. There's a certain resistance mechanism, but also for meropenem and vabapetam, no activity against OXA48, for instance, and other resistance mechanisms. So here you already see that mechanisms matter, so you have to know what is behind it, and sometimes have also molecular diagnostics to be able to actually show that. So to conclude, the antibiotics guide is currently being finalized. So all the people who have registered to this webinar will get the link once it's launched, so you can actually access it, and it's free access. So open access, it has a translation opportunity. It is a living guideline. So also please, if you use it and you come into things that you think are not very handy, or for your setting maybe need something different, well, either we can make something novel for the local context, but it's also sometimes there are maybe broader things that you think can be improved. So we can actually, over time, continue to improve it and make sure it's up to date and meeting the needs of the people using it. So comment on it, and we can adjust if we think it's really indeed an improvement. So we had to make some choices that sometimes for high resource settings may choose differently, but Hind will show more about that later on, actually at this moment. So a Q&A session will be at the end, and you can ask your questions. So for now, I want to introduce the antibiotics guide team, who have been really crucial in actually developing the content for the different chapters. So we have Hind Kadri. She's an ID specialist from the American University of Beirut, and she concentrated in her work on multi-drug resistant organisms, resistance, and stewardship. And she's now working as an ID specialist in Saudi Arabia. Then we have Neda Kostova. who is working at the Radboud University Medical Center. She's originally a pharmacist and a public health specialist with also a lot of experience in low resource settings in the field of AMR and stewardship, and an important driving force for instance, in developing the website for the antibiotics guide. And then Teske Schoffele, she recently joined the team. She's an ID specialist at Radboud University and was, for instance, recently involved with the AskMIT guideline on stewardship in the emergency department setting. And she's also coordinating various stewardship projects also in low resource settings. And without further ado, so Hind will be the one presenting what is in it and what is not in the antibiotics guide. So I will stop sharing and leave the floor to Hind. So Hind, the floor is all yours.
So thank you, Dr. Heiman, for a beautiful introduction. Thank you, Dr. Kanj and Dr. Heiman, for leading this project and giving me the opportunity to participate in this webinar. I also want to thank or express gratitude to my colleagues Neda and Teske for their contribution and amazing teamwork. Okay, so this will be an outline of my presentation today on how the guideline was developed and how to use it. So here's an overview on the most important steps we went through for the development of this guideline. So first of all, we wanted to have a better idea on the interest of clinicians, what they're expecting from these guidelines, and what they wanted to see. So we addressed this by questioning them through a stakeholder survey. Now, based on the results of the survey, this gave us a full-blown idea about the scope of interest and the needed topics for inclusion. So we started out with the most important chapters. We performed an extensive literature review. We compiled a large amount of evidence from the international guidelines, and we tailored it, depicted certain information in a way that's suitable for low-resource settings. Then we tabulated the data, followed by an initial review by our project team. And then this was done through multiple discussions and commentaries until we reached a final draft, an initial final draft. And then we had it externally reviewed by peer reviewing by independent experts in other societies. And again, the data was adjusted again by consensus to reach a final draft that could be implemented into the web-based platform. Then a series of webinars for promotion of our work was started today, and I'm happy here to be participating in the first one for global launching. So just taking a deeper look at the stakeholder survey, which was essential for paving the way to the rest of this project. So this survey was done because we wanted to know what clinicians wanted to see and what was important for them and their preferences for such a guideline. So the survey was sent to clinicians among ISAC and APOA societies and other societies and professionals worldwide. And this came back with important results showing us that most concerning infections or most wanted infections to be presented were the bloodstream infections, including endocarditis, respiratory infections, and neurological infections such as meningitis. And it's worth mentioning that more than half of the clinicians here were from low to middle income countries. And this is important because we were concentrating on the work. We were concentrating our work on the resource constraint settings. So this is why we were stressing whether they are from low to middle income countries or high income countries. In addition, we asked them other information on what these topics should include. So under each topic, we wanted several points to be presented, such as preferred and alternative therapies on different pathogens and based also on their susceptibilities. And we also asked them what's the preferred means of displaying this information, and most of them answered preferred to have a web-based platform or application. So regarding the development process, the stakeholder survey, it paved the way for the rest of the work. It provided a template and a set of headlines and major syndromes to start out with. So for each topic, we screened a good set of international guidelines with extensive review for each topic. Approximately, we had 5 guidelines for each topic. And for rare organisms, we depended on expert opinion or case series. We compiled all of this available evidence and tailored this data in an attempt to make it applicable when there's low resources. We took into consideration, of course, the cost, the ease of use, the availability of the drugs, and even the duration of therapy was sometimes adjusted. Now, in order to make it instrumental or relevant to contribute to the global goals for AMR and aligned with the WHO efforts, we made our antibiotics in accordance with the WHO access classification, and this is to stress on clinicians to use the access class of antibiotics. And this will be presented by Dr. Mike Sharland in the next presentation. Okay, so this is the general example or a drop down on how we thought when we developed these guidelines. This is like the thought process. So because they were pathogen directed, we started out with the common and uncommon pathogens for each chapter, and we moved according to susceptibility patterns. We proposed primary regimens, and then we discussed, is this locally available? Is this suitable for locally or low resource settings? Is this should this be given in an oral or an IV format? And then we chose an alternative regimen. Also, we had beta-lactam and non-beta-lactam alternatives. We had alternatives in case the IV form was not present, and we left a comment section to suggest IV form to oral switch, or in cases of comorbid conditions or immunocompromised patients, we had some comments on those. And we suggested some investigations in case they need to be monitored with the proposed regimens. So regarding the validation process, once we had an initial draft of the compiled data, we began with an internal review from our project team members under the guidance of Dr. Kanj and Dr. Wertheim. We had an extensive internal discussions, mainly weekly or even daily, and we shared comments and opinions, especially when there is a scarce data or on debatable issues. But our bold line was always to contextualize our approach to resource-limited settings. So after we reached a consensus for an initial draft, we sent it out for peer review by experts in the field. The majority were experts from ISAC or APOA, but members from other societies also participated, and we had at least 2 experts for each topic. And we took their comments into consideration. Adjustments were made in order to reach a final draft by consensus of the whole team. And we had this draft implemented or integrated into a web-based platform for pilot testing and then promotion for use worldwide. So here's the framework. Basically, this is how the web page will look. It's very simple, and it's divided based on organ site infections. On the left, there's a search box where you can search for a pathogen or a syndrome, and after which you'll get a drop-down panel showing you the susceptibilities. I'll show you an example. So this is, for example, hospital-acquired pneumonia. The pathogen is Staph aureus with a susceptibility to methicillin. So our primary regimen was nafcillin in this case. We also proposed the dose and the method of administration and the duration. And we proposed alternative regimens. Mainly, we concentrated on having a beta-lactam and a non-beta-lactam antibiotic in case of allergy. And we had some comments in the comment section for additional information. So it is pathogen directed, it's either friendly, it's free, and it's optimal for limited resources. So what are we looking for in the future regarding the ABX guide? So as you know, with the events happening around the world, the new and the emerging pathogens, the resistance mechanisms, the emerging syndromes, and with the advancement of antibiotic production in the pipelines and the updates on the guidance coming up frequently, we will be annually reviewing the data presented in these guidelines. We're also attempting at studying its applicability. So we want to know how well it worked with the hands of clinicians and maybe adjust it according to their on-ground experience and their feedback. Definitely, the ultimate goal is to study the effect of its implementation on AMR and its implications on encouraging antimicrobial stewardship. This is all part of the global action plan. And lastly, we look forward to performing a mobile application on the for the ABX guide and translating it into several languages. And with that, I can give an end to this presentation. Sincerely, thanking you for your attention. Thank you.
So it's now my distinct pleasure to introduce our next speaker, and we're very thankful for him joining us. And this is Professor Mike Sharland. He is a consultant in pediatric infectious diseases at St. George University Hospitals in London. Professor Sharland is a leading global expert in antibiotic use and policy. He is an advisor to the WHO on optimal antibiotic use, including he is a member of the expert committee on the selection and use of essential medicines. Dr. Sharland is the chair of the antibiotic working group that developed the AWARE project and the WHO AWARE book, and we're going to hear a lot about it. He leads the Wellcome Trust-funded ADILA project, developing novel risk-adjusted models for optimal AWARE antibiotic use. He also leads multiple global trials on improving antibiotic use in children. So Professor Sharland is going to talk to us about the WHO AWARE book. Mike, the microphone is yours.
Thank you very much indeed, and thank you for the opportunity for sort of talking today. This is an excellent and really needed initiative from sort of kind of from ISAC, and so we're very pleased to sort of have this discussion today in the webinar and sort of talk through how this sort of lines up, as Heiman was talking about, with the empiric sort of treatment guide in the WHO AWARE book. Very briefly, I'll just go back a little bit sort of kind of to history. The essential medicines list, of course, is the list that of what medicines should be available. This is the model list the WHO produces each year since 1977, and then different countries produce their own national essential medicines list, adapted to their own sort of requirements and their own sort of disease burden as well. So right from the very beginning in 1977, the first email had 16 antibiotics out of 240 medicines, and many of those, in fact, if you look, chloramphenicol, cloxacillin, gentamicin, are in fact really still the essential medicines even now, many years later as well, obviously. And so after the last sort of kind of General Assembly, the WHO was asked to sort of consider all of the antibiotics on the essential medicines, which has just been added on another one each year, to say, well, this is actually getting quite confusing, and can we try and put this into a more straightforward and simple way for stewardship and sort of monitoring and evaluation sort of purposes. So that in 2017, the WHO Essential Medicines Expert Committee then developed the AWARE classification of essential antibiotics. And so, this is and it's really designed, to be simple. So access antibiotics, narrow spectrum, ampicillin, penicillin, sort of first generation cephalosporins as well, and gentamicin. Watch more broad spectrum antibiotics used for more specific and limited infections, and they will group particularly in relation to their higher propensities for selecting resistance, of course, and reserve really as the last resort. So that's for enhanced sort of stewardship perspectives as well. And that's a little bit like Heiman was talking about as well, about the the sort of kind of all of these guidelines are in fact live documents, and this sort of a list is a live document because in the end, why antibiotics should be in one group rather than another depends really on in many ways on multiple factors, not only the sort of their clinical utility, but also their selection for resistance, but also their cost, their toxicity, their impact on the microbiome. So I think this is something that's evolving and will continue to evolve over time. There's a new category that's been added on, not recommended, which we won't talk about today. And the antibiotic, this was then extended on for antibiotics that are actually not on the essential medicines list, but multiple other antibiotics that are being used in humans globally actually were given an access or a watch or reserve categorization, if you like. There's 41 antibiotics on the essential medicines list, and there's now 270 antibiotics that actually have an AWARE group allocated to them. And this led into the target that of GPW that says that actually 60% of all total antibiotic use at a country level should be access antibiotics. There is that now going to be debated at the UN General Assembly on the 26th of September. The EU has pushed that up to 65%, and the UN General Assembly is debating a figure of 70%. And so we can come back and talk about that a little bit later. So I think the point about the medicines that we've got at the moment is that they are predominantly a mixture of sort of primary care and secondary care treatment. and oral and intravenous antibiotics. And so after some time, it was discussed that in fact it would be helpful to have a more overall guidance, of how antibiotics should be used across these different settings. WHO has specific antibiotic guidance for childhood pneumonia, for example, but actually there wasn't a sort of comprehensive uniform guide. And so it was agreed by the WHO this should be sort of taken forward, and this led on to the antibiotic book, which was launched in 2022, and we're now working our way through getting thrown to the next edition of that, and there's a sort of and there will be plans for a regular and complete update, for the book as well. So it moved on from a list, which is classically what the essential medicines list was, just a list of the medicines that you should have available, onto sort of how you should use those. And it was the point of clinical presentation. So the assumption there was that the physician, or clinician had seen the patient and that had taken a history and examined them, but there was not related to any live laboratory diagnostic. Though the whole book was then done in collaboration with the Central Diagnostics List to work out what diagnostics need to be done. So the mission, it was all about the clinical diagnosis, the decision, and then obviously provided information on the drug dose and duration. And the target audience really is all health professionals giving sort of antibiotics as well. And the principles of the AWARE book were that it could be relevant and educational at the same time, and therefore provided details on all the different clinical infections, which we don't really know, but covers probably about 95% of sort of clinical infection prescribing. And there's simple sort of infographics that you can see for children and for adults as well. There's obviously a strong focus on the low and middle income setting, and also a strong focus on access antibiotic use in primary care, with a particularly strong focus on access, increasing the use of access antibiotics, within the sort of kind of primary care setting. And the resistance patterns and how they should vary is complicated. This work was done in relationship to glass, obviously. And the guidance, there was guidance given for example, typhoid, where related to sort of macrolide resistance, for example, or quinolone resistance in different settings. But it should be noted and discussed that actually for the hospital setting, then the empiric guidance given for the sort of multiple infections, complicated urinary tract infections, for example, complicated infections, are across all settings at the moment, and because of the lack of sort of good quality information that be able to to inform how that should be sort of localized, and that I think is a step forward and another step going forward. So I think I aim to then led into quality improvement programs. I think there's now a AWARE antibiotic app, which is also quite important, and it also talks about rapid oral step down and predominantly also no antibiotic care for where there's minor sort of respiratory tract infection, and gives a sort of a very risk-based approach to prescribing as well. As I discussed, there's quite complex issues how you should adapt guidance for the varying patterns of resistance locally and nationally. And I think this is going to evolve and emerge over time. We have very little data, in fact, on the high quality data on the impact of concordance and discordance on sort of on mortality outcomes, predominantly, particularly in the lower middle income setting. And in the ADILA project, actually, Ben Cooper's group are now working through we can bring that back in discussion further, sort of kind of trying to reduce the minimum bias associated with many of those original concordance discordance studies by taking a sort of emulating target trials. Thinking through the sort of trying to understand what this outcomes associated with that. But clearly what we also need is good clinical AMR surveillance. And so there is we need to identify much better these models of antibiotic modified mortality. And I think that very much fits in with this sort of kind of, the ABX guide, because, we need to work out what actually is the impact of these guidance. And we also want, in the long term, from a policy perspective, is to work out what the impact of this concordance, discordance relevance models are, and therefore how that feeds into access to antibiotics, particularly reserve antibiotics in different settings. So I think the next step really is to sort of think through about the impact of low-risk settings and trying to identify and increase sort of access antibiotics. There's a lot of work, I think, going on now in terms of policy goals and how you can then use these guidance on policy goals. And the WHO is going to be publishing later on this year, we hope, having gone through a very large Delphi, RAND, UCLA methodology, quality indicators for antibiotic prescribing for both the primary and secondary care setting, again, with a whole lot of quantity metrics associated with that as well. And I think we do need to think through much more complicated, the sort of as the AWARE goes through, thinking through sort of other subclassifications. Watch is a very large and broad brush sort of kind of group. And so I think there's going to be evolving adaptations of that. And we look forward to sort of trying to think through how this can evolve both therapy and empiric therapy, and then work with ISAC about some sort of targeted therapy as well. So I'll stop there. I think this was just a kind of a quick whiz, and we really want to thank all the antibiotic working group and all the other departments and everybody else who has very helped to develop that. And I think this fits very well with sort of the work that we're very, very encouraged to see how this is all fitting well to fit with the work that we're discussing today. Thank you, Heiman.
Well, thank you, Mike, for an excellent overview of the AWARE handbook. We already saw some questions coming in, but so there will be a sort of a panel discussion at the end. So first we have Paul Newton. So thank you, Mike. And Paul, please start sharing your slides while I start to introduce you. So Professor Paul Newton is also a doctor in infectious diseases and a professor of tropical medicine in the Center for Tropical Medicine and Global Health at Oxford University in the United Kingdom. And in Laos, with his Lao colleagues, they founded the Lao Oxford Matsot Hospital Wellcome Trust Research Unit, LOMRU, within Matsot Hospital in Vientiane, and he was there the director up to 2019. And while doing so, they had a really great sustainable project and a team now really focusing on the quality of medicine, to answer key questions on the epidemiology, the impact and detection of substandard or even falsified medical products. And he's now doing that from Oxford University in the UK. His work includes alerting to the public health the dangers of poor quality medicines, conducting innovative forensic analysis to understand falsified medicine origins, evaluating promising portable devices for empowering drug inspectors, which is really important, and developed the first consensus guideline on the conduct and reporting of medicine quality surveys. And he also organized the first international conference and a yearly short course on medicine quality and public health, and built the multinational capacity for research to inform policy on prevention, detection, and response to substandard and falsified medical products. So a really crucial and important subject, and really look forward to hear your talk on the quality of antibiotics and the impact on patient outcome and drug resistance. So over to you, Paul, and thank you for joining.
Thank you very much, Heiman, for that introduction, and thank you for inviting me to discuss this neglected issue in this very interesting webinar. So the quality of antibiotics and patient outcome and AMR. So many of us on this webinar and globally are involved in many of those aspects of medical research on the left, to optimize national policy and to produce guidelines. But all that work is potentially negated if there's poor prescribing, poor adherence, and poor quality antimicrobials. That's sort of the final common pathway to what patients actually use. There's been lots of controversy in the past about the definitions of poor quality medicines, including antimicrobials. And in 2017, the WHO developed a consensus on a terminology from a public health perspective, not from an intellectual property perspective. So now in public health, we don't use the term counterfeit, we use the term falsified, which are medicines that are deliberately and fraudulently misrepresenting what they're supposed to be. So by definition, it's a criminal offense. And those who are making these, at least at high level, know what they're doing. In contrast to that, some standard medicines are those where there isn't criminal intent, but where there's errors in either manufacturing of medicines or in supply chains, such as degradation of medicines in supply chains if they are too hot or too cold, etc., etc. So substandard includes both degraded medicines and those that result from errors in factories. And since such errors will always happen, we're all human. And just as there are recalls of parts for spare parts for cars and aeroplanes, etc., then we will be faced with this issue for medicines, though we need to do all that we can to minimize that. There's a long history of this as an issue. First of all, for falsified medicines at the top left, that's a picture from the 12th century of physicians beating a pharmacist in what's now Saudi Arabia for selling them poor quality herbal medicines. There were multiple examples since then. One particularly difficult example was in post-war Europe when penicillin was available really for the first time outside of times of war, and there was a big trade in fake penicillin in the mid-1940s, as illustrated there from a newspaper report where a group of German and American soldiers sold more than $1 million worth of fake penicillin in Berlin. And this story led to the making of the film The Third Man, which some of you may have come across, set in post-war Vienna. And the picture in the middle is of the fictional intelligence officer with vials of fake penicillin on his desk. There have been multiple other examples, such as at the bottom left, falsified tetracycline made by a company called Brainy. But this was found in Cambodia, and there was no evidence that there was a company called Brainy in anywhere within the region. And so it was seemed to be a fake company making a fake tetracycline that contained no tetracycline, so it would have zero efficacy. WHO issue regular reports on this issue, and one example is the falsified coamoxiclav that was found in Uganda and Kenya a few years ago. And this again contained no coamoxiclav. There are also multiple examples of substandard antimicrobials. One of the key early ones was the disaster in the United States when a company in Tennessee used diethylene glycol to make a sulfonamide antibiotic syrup. But at that time, it wasn't known that diethylene glycol is a renal toxin, although there was some evidence because it was not allowed in food, but that was largely because there were no data to support its safety. But over 100 people died in the States from renal failure, and this had a very big impact within the States and led to the beefing up of the US FDA in 1938. Multiple other examples, this landmark study from Pakistan where they fascinatingly used the technique, if you like, an inverse antibiotic susceptibility testing, where they took ofloxacin available on the market, they made it into a liquid form, and they put this on agar plates of 3 different reference bacteria, and found that 4 of those brands had no activity whatsoever on fluoroquinolone-susceptible E. coli. So that was a very innovative way of demonstrating relatively simply that there was a problem with these products. And this wasn't by intent, it was an error in manufacturing. In our group, we're linked with the Infectious Disease Data Observatory, and together we built the Medicine Quality Scientific Literature Surveyor that tries to map all the global literature on the quality of medical products. And it's in English and in French. And the website is on the EDO website, and here at the bottom of this slide. This is a map that I extracted yesterday of the global reports from the scientific literature of the quality of antibiotics. As you can see, there are many reports globally. Red is not good, green is good, as illustrated on the scale in the middle. There's probably more reports which would be green, but probably unpublished. And crucially, this map does not include reports from medicine regulatory authorities that are not in the scientific literature or from the pharmaceutical industry. It just represents what's in the public domain scientific literature. But as you can see, there's still an awful lot of red points implying severe issues around the world. And if you wish to use this, you can make reports for a country particularly interests you, and you can download the data as well. We use this online system, the database behind that, to review the information we could find on the quality of antibiotics globally. And we found 106 surveys with about nearly 14,000 samples tested. Excuse me. But the median number of samples tested by survey was low at just 47. And there are lots of problems with the data behind this, poor quality and poor quantity. But this data set suggested that about 17% failed at least one of the quality tests used, with particularly affected amoxicillin, sulfamethoxytrimethoprim, and ciprofloxacin, with failure rates as listed there. Interestingly, we found no SF survey data for WHO reserve group. They were mostly in the access group. But most crucially, because the data behind this analysis are of poor quality, many of the studies were conducted using convenience rather than random surveying. This does not mean that 70% of global antibiotic supply is poor quality. Excuse me. Probably the vast majority use globally a good quality, but there are hotspots through time and space of severe issues. But would argue that even if 1% of the global antibiotic consumption was poor quality, that would have a significant public health impact. So what are the impact of substandard and falsified antimicrobials? WHO issued a very useful report in 2017, entitled at the top right, trying to list how these neglected public health issues impact on patients and on communities. Clearly, if you have an antibiotic that you expect to cure or ameliorate an infection, and it does not contain any of that antibiotic, the consequences will not be good. And there are natural experiments to try and understand, to help us understand what would happen to patient outcome by using dose per milligram body weight. This is analysis from the Worldwide Antimalarial Resistance Network of a Coartem, artemether lumefantrine for uncomplicated falciparum malaria, with the outcome as recrudescent at day 28. And as you can see, the recrudescent rate goes up significantly, as one would expect, at lower milligram per kilogram body weight dose. Therefore, suggesting that if one was of the normal standard body weight, but one had ingested an antimalarial that had less than the expected stated amount of active ingredient, that your recrudescent rate would be higher. So that's in terms of patient outcome for malaria, but there'll be very few analyses for antibiotics. WHO and the University of Edinburgh included in the report at top right the statement in brown at the bottom on if there was a 10% prevalence, which is the WHO estimate for the global prevalence of substandard or falsified medicines, estimates a very significant increase in childhood pneumonia deaths. But again, this is based on relatively poor quality evidence. So how important are poor quality medicines as drivers of AMR? It's logical that those producing drug levels within the window of selection will engender AMR, but those with no active ingredient, apart from which is quite common in falsified medicines, with a few exceptions, would thought to be unlikely to be able to drive AMR. But that still may be the case for malaria through the mechanism of hyperparasitemia. Alison Holmes and colleagues wrote a paper in the Lancet trying to look at the different drivers of antimicrobial resistance, and suboptimal dosing, which includes substandard or falsified medicines, is the light blue circle in the middle. So low to moderate evidence that it contributes to AMR, but a moderate risk of it contributing as a driver. But this again is largely evidence-based. It was based on people's opinions. But a key thing will be where antimicrobials are, where the pathogens are, and where antimicrobial resistance is first happening and how it spreads. These are some of the recent papers on this issue. There's been very little laboratory work on this, largely from Mohammed Zaman's group in Boston University in the US. And Nick White has led on arguing that hyperparasitemia and low dosing would be important drivers of antimalarial drug resistance. Sean Cavani and Ben Cooper in Oxford and ourselves wrote a review of this issue a few months ago. This is the references there at the top left. One often neglected issue is that falsified antimalarials and sometimes substandard antimalarials contain wrong, unexpected active ingredients at top right on this slide, including, for example, ciprofloxacin and chloramphenicol in falsified antimalarials. And so this will lead to the cryptic exposure of patients, and hence the bacteria that they're infected with or carrying being exposed to antibiotics that conventional surveillance would not pick up as exposed. Sean worked on this modeling, which is illustrated in his paper, looking at the consequences of different dose in relation to percentage active pharmaceutical ingredient or API on the risk of resistance emergence. But the distinction between substandard and falsified, although really important for how to intervene, whether it needs legal criminal investigation or whether it needs regulatory investigation, is not dissolution profile of that, determining the bioavailability. And the key aspect, which has not really been explored in much detail yet, are the pharmacokinetic, pharmacodynamic relationships, which will help inform the theoretical impact of different percentage API dissolution profiles on the engendering of drug resistance. And the Wellcome Trust funded FOREST-PHAR and the Fleming Fund funded GRAM projects are investigating this, led by Ben Cooper and Christian Dolacek in Oxford and Andy Stakakis in Washington. Thank you very much.
Thank you very much, Paul, for your excellent presentation. We have received several questions. I think I'll try to ask these questions, alternating between myself and Heiman as moderating of this Q&A session. So I'm going to start with some of the questions we received online. The first one, it says, by Dr. Shaista Banu, the presentation are very good. I just wonder how long the guideline will be applicable because resistance and pathogens emerge time by time. Heiman, can you address this?
Yeah, certainly. So it's a great question. So as I tried to point out, it's really a living guideline. So it will be continuously updated. So ISAC has committed to continue the funding to actually keep it updated and have an editorial board monitoring it. And there's a commentary section. So people who find that there are certain developments that make certain things out of date. So please continue to give us feedback so we can also take notice of that, see if it's indeed the right sort of comment, and we can adjust if we think it's needed. And yeah, of course, it depends on certain urgency. And of course, always discussions about certain dosing. And so we sometimes have to wait a little bit until all the experts agree on a certain dose before something is updated. But in the end, yeah, there is an ongoing editorial board and we'll be updating it along the way. Over.
I just would like to ask on actually to mention something that perhaps Hind wanted to expand on, that sometimes when we have the choice between several drugs within the same class, we always try to pick the drug that was the narrowest spectrum. So for example, if we had to pick quinolones, among all the quinolones that are available, the choice was for that particular pathogen, pick the one that is the narrowest spectrum and perhaps the one that has the least side effect profile. I'm sure some other questions about some practical things will come up in the questions by the audience. Just a question, I think, Heiman, another one, since we have you here, they're asking about the modalities used for stakeholder survey dissemination.
Yeah, so we use the ISAC newsletter, the whole network, email lists, and also via personal communication and snowballing to get the survey. And also in AskMed Amsterdam, actually, that's sort of where it started. We had various other stakeholder groups actually having a discussion we had at AskMed in Amsterdam at the time. But mainly, yeah, the survey was through snowballing and through the ISAC network.
So a question for Mike from Joseph Odior. So he's asking, what is your take on co-therapy of antibiotics and bacteriophages to tackle tough drug-resistant pathogenic bacteria?
I think it's very interesting about emerging sort of kind of treatment options, of course. The Aware book provides guidance on books that are on medicines that are actually on the essential medicines list. So that's a complicated process, obviously. And there are only 41 out of 270 antibiotics, as I discussed, that are actually on the essential medicines list. Clearly, they need to be licensed, of course, and also to be sort of kind of and widely available, obviously, as well. So I think medicine, what will be on the ML and what will be used in routine care will emerge and will change, quite markedly over the next sort of decade or so. Hopefully. And we look forward to evaluating all the clinical trials, sort of kind of evidence as that comes through. I think we have to recognize that this is empiric treatment, for the AWARE book, and it's empiric treatment, not targeted treatment. And obviously, in many of the countries, that the book is used, of course, there are massive resource issues for healthcare budgets, with around a third of people globally, I think, now living in countries where debt repayment is, significantly impacting on the healthcare budget as well. So, of course, so I was talking about cost and availability is going to be one of the key features, I think, in terms of treatment going forward. Thanks.
Yeah, just to add, so if anyone is interested in reading a non-medical book about bacteriophage, there is that book that I read recently called The Perfect Predator. Scientist race with antimicrobial resistance. It tells the story of a physician who acquired MDR Acinetobacter while in Egypt, and he was about to really pass away until they got him the bacteriophage from the US. And so they tell the story about how this was done. But I tell you, last year we tried to acquire a bacteriophage for a patient who had difficult to treat Pseudomonas aeruginosa pneumonia that was resistant to everything, including the novel drugs. And the cost is really prohibitive. But many laboratories in various countries are developing their own bacteriophages. So we eagerly await to see what will happen and in terms of accessibility of these. Heiman, would you like to ask the next question?
Yeah, so just a sort of a note about the certificates people have been asking. So that's more a general comment, is that people attending for the majority of the webinar, they will get a certificate by email. So I think there have been some other questions also for Mike and one maybe for myself. So maybe to start about the key challenges about implementation the antibiotics guide in resource-constrained settings, from Maya Dager, with a limited access to diagnostic tools and lab facilities. So indeed, that's the case. Actually, we see this also, the antibiotics guide really as an inspiration to actually see, okay, it really actually matters. I think in areas where there's more complex kind of patients, so I think in a small clinic, I think the directed therapy guide will not be very useful and probably too much and not proportionate to the issue. And the AWARE guide will be sufficiently, sufficient to treat the patients. But I think in settings, larger hospitals, which every country has, with more complex patients, with complex surgeries, prosthetic infections, transplantation, I mean, you do see also in all kinds of situations, also in middle-income countries, these things happening. And I think really there are sufficient resources in the end that should be allocated to more diagnostics and directed therapies. And I think with the whole push of AMR and the UN Assembly, there's actually more attention to it now. So I think one is it will take time, and that will be to keep on informing people about the antibiotics guide. And directed therapy actually improves outcome, and you need diagnostics for it in that sense to inspire, but more in the bigger facilities. And the other challenge will be language. So we want to promote people to take ownership of the antibiotics guide. So it's really open access, so people can actually translate. And we're looking with the company to build the website to allow for maybe artificial intelligence tools to actually to support that. So that is my answer. So I think then the next question is from Halis Akalin. So maybe this one is for Paul. So thank you for the nice presentations. Can you solve AMR problem without controlling antibiotic use in animals and agriculture? So maybe you want to reflect on that. And I remember we had these kinds of discussions also with the substandard meeting, phosphate and medicine meeting we had in Oxford a couple of months ago. So over to you, Paul.
Thank you, Heiman. Thank you, Hannah, for the question. I don't see how we can address this problem without addressing antibiotic use in animals and agriculture. A key part, and there needs to be much more engagement. I think WOHA, the World Organization of Animal Health, has now have a interesting program on this, linking up with WHO. But I think one needs to divide antibiotic use in agriculture between those that are for treating disease in animals and those that are used for increasing production, and they need to be viewed through in different ways, reducing those that are used for production, and trying to optimize use for those antibiotics being used to treat diseased livestock and also pets as well. Would Heiman and Mike and Souha and Hind agree with that?
So, Mike, any additions?
No, fully agree. And I think the other point about this, Paul, is around manufacturing, because many, much of the global manufacturing of antibiotics, of course, many manufacturers will manufacture antibiotics for humans and animals and the same plants, and so using sort of and so I think there's much further work to be done to sort of think through the sort of whole supply chain, and in many ways, I think, we need to start to think about antimicrobial resistance really as a medicines management issue, in my view, and now start to engage much more with procurement and sort of and the pharmaceutical supply chains and as well, because we to control sort of antimicrobial use in the animal and the human supply chain, then we need to go further back upstream to have a look at the original sort of supply chains and manufacturing chains as well. Yeah, agree, finally. Agree indeed.
So, Hind, any further additions from your end regarding that question, the agricultural one? We don't hear you yet. I think you're still mute. We don't hear you. So,
so while Hind is getting her microphone to work, I have a question for Paul. So whenever various hospitals, academic centers try to introduce generic drugs into their formulary, there's always a list of like 20, 25 different factors that the drug has to satisfy before accepting a generic on board. And obviously, a lot of people are now moving towards generic to save money. So my question to you is, if you have to pick 3 or 4 essential factors that are more important than anything else to allow a generic antibiotic to be used in a hospital setting, can you share your thoughts on what do you think are the most essential? Because sometimes when you have a huge list with too many details, it becomes difficult to implement.
Thank you, Souha. I think a key thing is that the antibiotics used, whether they're generic or innovative, are registered with the relevant national medicines regulatory authority. In quite a lot of countries around the world, quite a lot of the medicine supply may not be registered and therefore evaluated by the National Medicines Regulatory Authority. So I think one needs to check with the NMRA, the National Medicine Regulatory Authority list, to make sure that they are approved for use in the country by that particular manufacturer. And to be wary of, in terms of substandard medicines, to keep an eye on international alerts, on WHO alerts, on alerts from adjoining countries or in the same region that there might be issues. And this spans both innovative and generic. And then for falsified medicines, again, to keep an eye on the WHO alert system and press reports from the country within one is in, and also countries within the region for any rumors of issues. And also to beware of, look for, when one receives the medicines or has a sample, to check for typos, inconsistencies, and that they're not unexpectedly inexpensive. There's only so, it's a very competitive market, and there's only a certain lower limit that you can go down to, that it's possible to make something that's good quality. So to be aware of something that looks too good a deal, that is it really possible for a manufacturer to make drug X for that amount? And if it is lower than one might expect, to have a further think about that. Is that, have corners been cut? And all of this applies to both generic and the innovative industry. It's does that answer your question?
Absolutely. Thank you. I'm gonna answer a very simple question and then pass the microphone to Heiman. So Goyitom is asking, what's new in the antibiotic guide compared to the AWARE handbook of WHO? I just want to reiterate on the fact that they are really not the same. The WHO AWARE book is an empirical therapy book. The ABX guide is pathogen-directed, as Hind explained, depending on which organism, the site of infection, etc. So they're really different. So just to clarify that in case you logged in the presentation with a bit of a delay. Heiman.
Yeah, thank you. So another question from Teklu Gebre Michael for Mike. So why the AWARE does not include antifungals? Can you say a bit about that?
Yeah, so I mean, as discussed before, a lot of this is to do with development over time, and the WHO, of course, now are taking on a whole sort of program of work related to sort of antifungals, obviously, and in particular, you know, obviously, so there's the fungal priority pathogen list, and now sort of kind of thinking through the antifungal medicines. So that is in the in the work stream. I think one also has to be recognized that actually, you know, there is going to be a need to, well, we need to think through the sort of classification of that, quite complicated, and I think it's thinking through what I was discussing, really the factors associated with why different medicines will be in different sort of groups. So yes, it's in the game, it's in the work plan, and but that clearly is something that also it needs a really careful structure as to what will be taken forward. Thanks.
Yeah, so maybe Hind, if you are there now. So antifungals, something you think should be considered for the antibiotics guide, so it should then become maybe more antimicrobial guide, but that's beside the point. But any thoughts on that from you, Hind?
Are you hearing me now?
Yes, yeah, loud and clear.
Yeah, I would like to probably prepare the guidelines on resistant antifungals, given that we have like the rise of Candida auris right now.
Yeah,
also,
to show how also different Candida or other fungal species can actually also impact the choice of your antibiotics. Also, their diagnostics can be quite crucial in resistance testing. So yeah, so thank you for that. And Paul, any knowledge of falsified or substandard quality antifungals? Anybody doing that, something in that area?
No, I haven't come across anybody working on that. I'm aware of a few case reports from the distant past, but I don't remember anything recently. But I think it probably, I fear it's because people have not looked. Mostly when we look, I'm afraid that we find.
Yes, well, I think antifungals and fungal disease are getting more traction now. So I think probably soon we'll hear more probably about it. So maybe something for a Hind and Mike to discuss. So we've been a lot of issues regarding indeed what also Souha referred to, like with the ciprofloxacin, and which one do you choose? There's so many different kinds, but also with Staph aureus, do you choose nafcillin or cloxacillin or flucloxacillin? Which one do we show? So maybe Mike, what are your thoughts on that? Or how, was there some strategy you think should be recommended, or and how did Aware deal with that?
Yeah, so this is obviously it's an essential medicines list issue. So of course the EML has a way of designating sort of equivalents, and so medicines for any drug that goes onto the EML, there's an assessment that actually, and if you go onto the EML, which you can see, and I'll put the link in the chat, but then you can see all the other drugs that are regarded as equivalent. They're sort of kind of that's equivalent on the basis of clinical efficacy. I mean, and so I think that then becomes quite complicated when we try and take into account all the other different components. So, as part of the work stream for the Aware book, we're just working our way through an enormous, or the Italian colleagues are working through an enormous systematic review of the toxicity associated with different antibiotics, and also the impact of the microbiome. And so, hopefully we'll be able to then try and think through the different different impacts. I think in fact that actually becomes quite complicated, because different countries may wish to make different decisions based on the sort of on their own settings. So it is potentially possible that cost obviously is critically important for whether patients can or cannot afford an antibiotic, and and in some settings then obviously an antibiotic that is available is actually the one that you would use because it's available, as opposed to a drug that may have less impact on resistance and may act in that. So I think it's I can see this evolving over time, and I can see this heading towards really taking into account not only the sort of the microbiological factors, but actually adding in other factors in terms of toxicity, cost, and availability, sort of into the decision-making process as time goes on. Thanks.
Yeah, this really sort of reflects the discussions that we had quite extensively. So, Hind, maybe your thoughts or any wise words in that area? Over to you.
I'm thinking as much as we had probably some difficulty or debate in discussing the antimicrobials in low resource settings, how difficult it would be for the when we're talking about fungal resistance. So I'm not sure about the with the maybe less data compared to the evident guidelines on antimicrobials, how much how applicable that's going to be into the ABX guide.
Yeah, so it's
always, well, we have to think about the very limited resources in the diagnostics, even about the methods of resistance, because in when we were talking about the bacterial resistance, we were even discussing if we will see the beta-lactamases and how they were, if we have them in the low resource settings, if we know even if it's KPCs or OXA48, we were very skeptical about that. So I'm not sure about the fungal resistance, if we're going to be able to reach that part in the resistance pattern they have.
Yeah, I mean, it's sort of, referring back to the old discussion, but so thanks for that addition. So maybe for me, last question before I hand over to Souha for Paul. So regarding the substandard of falsified medicines, I mean, is there something that you think, like maybe a Aware handbook or maybe the antibiotics guide, should we, for certain specific antibiotics which you think are high risk for being substandard or falsified, should we give some advice or in a comment, something about how to recognize substandard or falsified? Any thoughts on that or any advice in that area?
Was that addressed to me, Heiman?
Yes, to you, Paul. Yeah.
Thanks. I think it will be difficult to highlight particular antibiotics at risk because it changes through time and space. But I think a more general introduction to the issue with things that we discussed earlier about unexpectedly low cost and typos and other such things would be useful to include. But also, what do people do if they are uncertain? Advice about what to do, such as asking the pharmacist, discussing with the medicine regulatory authority for that area, and also the wonderful WHO reporting system. You can send an email to rapidalert.who.org, I think, to report any issues that you may have.
Yeah, that's an excellent suggestion. So I think actually that's a good idea, and we'll certainly consider to actually have a link or some information about that. So, so thanks for that. So over to you, Souha, to maybe final questions and a wrap up.
Yeah, thank you, Heiman. So there were a couple of questions about where can we find the link for the ABX guide. I think once it is ready, you will find it on the ISAC website, but also all people who have registered for the webinar will receive them. And there was another question about where can we find the link for this webinar, and this will be found on the ISAC website under ISAC Academy, but also on the ISAC YouTube channel. So perhaps I have a question for Mike, and that is a question that perhaps we can learn from, that we can also adopt for the ABX guide. And that is, if you have had any data on in the countries where the AWARE book has been adapted, do you have any measurable outcome in terms of reducing antibiotic cost or AMR? Have you had any data collected to be able to have analysis?
No, and the answer to that is, for very clear and sort of kind of obvious reasons there of the complexity of antimicrobial resistance and the multiple sort of routes through and the modality we're discussing with animal use and elsewhere. So I think the only thing that the data that we are collecting and we can collect are sort of use data. Obviously, we also have the app use the data. The book is being translated, globally into multiple languages across, Latin America and China and Russia and Francophone and multiple other countries. Also, we know that like any other WHO guideline, then some countries have just almost adopted that as their national antibiotic treatment guideline. Other countries have gone through a very detailed review process and adapted that slightly in relationship to antibiotic access, as we talked about, with particularly in relationship to their sort of national EML. So I think the metrics that we can do in the future are going to be related to the are going to be related to the sort of use type metrics. But the book, basically the AWARE system is much larger than the book. So I think the book is only a small part of the AWARE system. This is now the way in which WHO is trying to consider optimal antibiotic use. I think we now have to consider rather than not only relative-based targets, 60%, 70% targets, but in the future, I think we're now going to be talking about volumes of antibiotic use based on your own local disease preference. So clearly in the future, antibiotic use at a national level should be based on your own population health needs, based on your burden of disease, your country's antimicrobial resistance data, what drugs are available, and multiple other factors associated with that. So that will, I think, lead on. It is most potentially possible that there will be an independent panel of AMR agreed at the UN General Assembly at the 26th of September. And it will be that independent panel that will start to sort of take forward policy interventions and a range of policy interventions that will then be looking at countries. And then, of course, the question is to how microbiology surveillance can impact on that. So I think things like the AbxGuide and the AWARE book are sort of adjuncts to policy, and antibiotic stewardship is incredibly important and incredibly necessary. And equally, we do need to not only have antibiotic stewardship in the secondary care setting, but we also need national policy, which includes all of the issues we're talking about, which is procurement and sort of pharmaceutical policies. And if we have those targets, those are the ones that we now need to have a look at and see what the impact on antimicrobial resistance are. So these guides are part of that process, but they're an adjunct to that sort of process. But I think they lead into the concept of help with national antibiotic policy implementation is the way we should be seeing this in the future. Thanks.
Yeah, thank you very much, Mike. a question to the microbiologist among us. Heiman, there is a question by Joseph Odior. How do we handle the poor antibiogram testing procedures in low-resource countries, which is in one way the contributor to the development of MDR strains? So how do you handle poor antibiograms?
One is to have them point out to the webinar of Fred Tenover in his e-learning module, but no, this is a good point. So the quality issue, and that's of course also for medicines, which can also have a poor quality, but indeed also for testing of drug resistance, getting more complicated due to rising resistance. You also have molecular methodologies. For some things you need microdilution, other things you need agar testing. So it's getting actually quite complex and confusing even. So I think one is that the European Union, you have UCAS with quite good guidelines and breakpoints and actually good suggestions for testing. But then, of course, you need the knowledge and the resources and then also the materials. So often the reagents that you need are not available in these settings. So the positive thing is with the UN Assembly and WHO, there's a really huge push now, and also in the latest Lancet series, that diagnostics are quite key and a fundamental pillar for AMR management. So we do hope that it's getting more attention and it really will take more time, and that also sort of basic microbiology is coming more to the picture, and hopefully these testing will become more routine and to a good standard, and also a standard that they can prove with actually participating in quality assessments and proficiency panel testing. So I think overall the investments are getting bigger in this area. So there's a good and positive hope, but it will take time.
All right, there are a couple of more questions, but I don't think we have time. There is a question about probiotics and use of broad spectrum antibiotics. This will be the topic of a webinar on its own, because I think there is some emerging data on the use of probiotics. Hopefully we can address this in future ISAC webinars. If you have any ideas that you'd like to listen to, please send your suggestion to Fee at the provided email at the beginning of this. I think with this, we're reminded by Fee that the next webinar is the Middle East, North Africa region, because in the regional webinars, we will try to address issues that are of interest to the particular region. And the next one is on September 17, and Fee shared the link to register for that webinar. And with that, I pass it on to Heiman to conclude.
Well, thank you, Souha. So yeah, it was an excellent webinar. So great thanks to Hind, Mike, and Paul for really great presentations, and also in nice answering the questions that the audience and also other panelists had. And Souha, was wonderful adventure with you, and we look forward to soon to provide people the link to the website, and look forward to get comments, either positive or negative. We want them all so we can improve it along the way. So thank you, and have a nice day.